Thank you for the opportunity to present today. Our mission is to reduce the treatment burden for patients that have ocular diseases. The first approach that we've taken to that is actually a depot formulation of a long-acting formulation that will allow us to treat potentially twice per year. We call it GB102. It targets multiple VEGF pathways, and we're currently in the clinic testing this program. I'll give a brief update on that today. In addition, we've now reformulated that product, and we'll give an update today on the fact that we can now formulate up to once a year treatment, and we have data to support that. We presented at ARVO recently. In addition, we're expanding further into other ocular indications, including glaucoma, where again, applying this novel technology to reduce treatment burden for patients with glaucoma. The technology originally came out of the Wilmer Eye Institute at Johns Hopkins to overcome some key challenges with many of the depot delivery platform technologies. First of which is to really reduce inflammation in the eye, and we're successfully achieving that preclinically, and we'll hope to share the clinical results later this year. The way that we do this is a proprietary technology of hydrophilic coating on PLGA microparticles. So PLGA has been around for many decades, as most of you know. The drug that we've selected as first into man is sunitinib, which blocks multiple VEGF pathways. And once we incorporate this into these small 25 micron PLGA microparticles, we then inject it into the vitreous. The particles aggregate together, so that's another part of our novel technology, is to have the particles aggregate and form a depot out of the visual axis. As you can see on the cartoon at the bottom of the slide, once we've injected this into a mini pig eye, it forms a nice depot implant, and then over time, as the PLGA degrades just naturally, it gets smaller and gets completely resorbed in the eye. The size of a 50 microliter injection in a human eye is approximately 5 millimeters, which is shown in the cartoon in the bottom right. I've previously showed some of this data. I just want to remind you that the pharmacokinetic profile that we had preclinically suggests that we should be able to achieve a 6-month dose duration in patients. In this study, we gave a single injection of GB102 to rabbits, and we saw peak levels achieved after about 3 to 4 months, at which point the depot is completely dissolved or resorbed. The unique attributes of this particular molecule that we're using, sunitinib, is it binds avidly to melanin. And so we see a continued release from the melanin granules in the back of the eye. And you can see very high levels in the RPE choroid and the retina of the target tissues that are sustained out to at least 6 months. The next question then becomes, what is the durability of the response? And so we've worked very closely with Peter Campochero's lab at the Johns Hopkins University and done this detailed study. So let me just walk you through this briefly. What we did is we injected a very large number of mice, initially either with Eylea or with our product or with placebo microparticles of our product, and then looked at the durability of the response by inducing laser in separate groups of these animals for choroidal neovascularization. You can see with Eylea, after 1 week, we see a very robust response. This has been published previously, so not unexpected that you'd see that with Eylea in this model. But by 9 weeks, you can see that that effect of Eylea or aflibercept is completely worn off back to placebo levels and no longer inhibits CNV formation. However, with 2 different doses of our drug, we see that it's very sustained, durable response out to at least 6 months in the high dose, and that those doses, once correlated to the equivalent doses that were used for Eylea in this study, are at or near the doses that we're testing currently in the clinic. So we do expect to see a nice sustained effect in patients. The ongoing study is outlined here. It's a 2-part study. The first part is a single ascending dose study. We've completed enrollment of 32 patients in this study. It's very important for me to highlight that these are not naive patients. So you're typically looking at naive patient data. These are patients that have already received an anti-VEGF. They've had at least 3 injections of an anti-VEGF therapy, and they've shown a response. When they would normally come back in for their next injection, they have fluid present, so they're having recurring fluid in the retina. We dose these patients with a single injection of our drug. And then monthly they come back and we look for fluid by OCT, we look for visual acuity checks, and obviously for safety. And so this study is currently ongoing, and we hope to be able to show the durability of the response that I just highlighted in animals in humans later this year. Once this part of the study is completed, we will select 2 different dose levels to go head-to-head in a non-inferiority study design against Eylea. This will be a randomized controlled study and double masked to allow us to achieve potential efficacy demonstration at 9 months in this study. Just briefly, the next generation of this formulation, as I mentioned in the beginning, GB103, this is data we shared at ARVO. You can see the duration here on the left-hand side, a single injection to rabbit. The depot lasts and delivers the drug for at least 10 months in these animals. And you can see that the levels in the RPE choroid are sustained well above the target levels that are necessary to inhibit CNV formation. And again, because of the melanin binding, we think this drug profile will last for at least 12 months, if not longer. So just briefly to wrap up our milestones, we hope to be able to share some of the initial data from the clinical program that I just outlined for GB102, the single ascending dose study later this year. And then we'll be starting off next year sometime on the part 2 of the study. In addition, as I mentioned, we have a glaucoma program that we're working on as well. This will be, again, a chance for us to reduce treatment burden in patients with glaucoma with at least a profile of every 6 months, if not longer. And we hope to be able to start this in the second half of next year. Thank you very much.